everyone, I'm Isabel, and today I'm going to talk about the technologies involved in medical diagnosis. So, what's medical diagnosis? I believe everyone's familiar with the concept. Medical diagnosis is the process of determining which disease or condition explains a person's symptoms and signs. Often, one or more diagnostic tests are involved during the process, including X-rays, biopsies, pregnancy tests, medical histories, and results from physical examination. So now I'm going to talk about some examples of use of diagnostic tests in some conditions. First, leukemia, a blood disease, and there are many diagnostic tests for this disease. First, the doctor will ask about medical history and conduct a physical exam. During the exam, the doctor will check for signs of disease, such as lumps, swelling in the lymph nodes, spleen, and liver, or anything else that seems unusual. Second, the doctor will need to do blood tests that check the levels and types of blood cells and look for changes in the shape of blood cells. But even if blood tests suggest leukemia, the doctor may look for signs of leukemia in the bone marrow by doing biopsy before making a diagnosis. Then, if indeed the leukemia cells are found in the bone marrow sample, the doctor may perform more tests like spinal tap and CT scan, ultrasound to determine if the disease has spread to other parts of the body. With a spinal tap, the doctor collects a sample of the fluid around the brain and spinal cord, and the CT scans and ultrasounds produce pictures of the inside of your body. Now, I want to talk to you about lactose intolerance, the latest diagnosis of which involves DNA diagnostic technology, which I think is pretty cool. So, most people in the world are lactose intolerant, but a genetic mutation where a cytosine nucleotide in a person's DNA is replaced with a thymine nucleotide allows them to digest milk. Previous tests for lactose intolerance were time-consuming and invasive, but the latest test determines whether a subject has the cytosine or thymine nucleotide in their DNA close to the lactase gene pretty quickly using the DNA diagnostic technology. Let's check them out. So first, a small amount of blood is taken from a test subject. Second, the subject's DNA is amplified using PCR, polymerase chain reaction. PCR uses primers to amplify the specific region of DNA that contains the single nucleotide polymorphism. Third, the restriction enzyme will cut the mutant DNA but leave the normal DNA intact. And then, the DNA fragments are separated by size using gel electrophoresis. Last, the pattern of DNA fragments indicate whether a person is lactose intolerant or lactose tolerant. Next, I want to briefly talk about Alzheimer's disease because I think the diagnostic tools are pretty fascinating and diverse. So doctors typically rely on the following two types of tests. Your doctor will perform a physical exam and is likely to check your overall neurological health by testing your reflexes, muscle tone and strength, ability to get up from the chair and walk across the room, sense of light and hearing, coordination and balance. Second, brain imaging. Brain imaging technology include magnetic resonance imaging, abbreviated MRI. An MRI uses radio waves and a strong magnetic field to produce detailed images of your brain. And a CT scan, which produces cross-sectional images of your brain. And A PET scan. During a PET scan, you will be injected in a vein with a low-level radioactive tracer. The tracer may be a special form of a glucose that shows overall activity in various brain regions. This can show which parts of your brain aren't functioning well. Last, cerebrospinal fluid. The spinal fluid can be tested for biomarkers that indicate the likelihood of Alzheimer's disease. So after all that, let me tell you exciting news about diagnostic technology breakthrough, published in an article from a website called Medical Use Today on December 2015. So one of the most commonly ordered blood tests is the complete blood cell count. Currently, the gold standard for the test requires the use of a lab with bulky and expensive equipment. But now, a new biosensor based on the microfluidic biochip promises to cut the need for a lab and bring the test to a patient's bedside. The innovation could make the diagnosis and screening of hundreds of diseases and treatments faster, easier, and cheaper. In their paper, the researchers describe how their biosensor yields counts of red blood cells, platelets, and white blood cells from only 11 microliters of blood. Yes, only 11 microliters of blood. The biosensor electrically counts the different types of blood cells based on their size and membrane properties. For the white blood cell and differential count, the device uses 10 microliters of blood. It picks out and destroys red blood cells and individually counts the remaining white blood cells. Then, specific types of white blood cells, for example, neutrophils, are counted using multi-frequency analysis, which is sensitive to different membrane properties. For the red blood cell and platelet count, the biosensor uses 1 microliter of whole blood. After the sample is diluted with saline on the chip, the cells are counted electrically. The total time to obtain the results is under 20 minutes. Wow, that's pretty short. And one of the researchers remarked, the translation of our technology will result in minimum to no experience requirement for device operation. Even patients can perform the test at the comfort of their home and share the results with their primary care physicians via electronic means too. The team is already developing a portable prototype of the cell counter, and the base unit of the reader will likely be handheld and will take cartridges about the size of a credit card. And the cost of the test will be around $10 instead of the $100 that the current test costs. And thank you. This is the end of my presentation. I really hope that you learned something interesting about my presentation. At least I learned many really interesting things about my presentation. Thank you. Here's the MLA citations for my presentation. Thank you.